Working mechanism of engineered virus vectors as vaccines. The core function of an engineered viral vector vaccine is to use the virus's natural ability to invade a cell as a delivery system for genetic code. This process instructs the body's own cells to become antigen factories, which then provokes a strong and diverse immune response. Step-by-step mechanism. 1. Delivery, infection/transduction. The vaccine is administered, usually via intramuscular injection. The engineered viral vector, example an adenovirus, travels through the bloodstream and uses its surface proteins to bind to receptors on the host cell, muscle cells, dendritic cells, or other immune cells. The vector then enters the cell and releases its genetic cargo, a DNA sequence containing the blueprint for the target pathogen's antigen, into the cell's nucleus. 2. Antigen production, the factory stage. The cell's own machinery transcribes the foreign DNA into messenger RNA, mRNA. The mRNA leaves the nucleus and is then translated by the cell's ribosomes into the target pathogen's antigen protein, example the SARS-CoV-2 spike protein. Crucially, since the vector's replication genes were removed, the virus cannot multiply or cause disease, it only serves to express the antigen. 3. Immune system activation, the teaching stage. The newly produced antigen is processed by the cell and presented to the immune system in 2 key ways, leading to both humoral, antibodies, and cellular, T-cell, immunity. Immune response. Mechanism. Result. Humoral immunity. The antigen protein is secreted by the cell or displayed on the cell surface, where it is detected by B cells. B cells mature into plasma cells that produce neutralizing antibodies. These antibodies circulate in the blood to block the actual virus from infecting cells. Cellular immunity. The cell's internal processing machinery loads fragments of the antigen onto its surface display molecules, MHCI. This flags the cell as producing a foreign protein. Cytotoxic T lymphocytes, CD8 plus T cells, recognize the foreign fragments and kill the cell. This is vital for destroying any cells infected by the actual pathogen in the future. 4. Memory formation. After the initial infection is cleared, specialized memory B cells and memory T cells remain in the body. Upon later exposure to the actual pathogen, these memory cells rapidly launch a massive, targeted defense, preventing severe disease. The viral vector effectively mimics a natural infection without causing illness, which is why it typically generates a robust and long-lasting cell-mediated, T-cell, immune response, a distinct advantage over many traditional subunit vaccines. Viral vector vaccines offer a powerful combination of genetic vaccine technology and the delivery mechanism of a virus. This results in specific advantages and limitations compared to other vaccine types. Advantages and limitations of viral vector vaccines. Advantages. Feature. Description. Robust and diverse immunity. They induce both a strong antibody response, humoral immunity, and an excellent T-cell response, cellular immunity. T-cell immunity is crucial for destroying infected cells and is often more robust than that generated by traditional or subunit vaccines. Mimic natural infection. By delivering genetic material directly inside the cell, they mimic what happens during a real viral infection, which naturally generates a highly effective and long-lasting immune memory. No adjuvant needed. The viral vector itself naturally activates the body's innate immune system, functioning as an internal adjuvant, meaning separate immune-boosting chemicals are often not required. High stability, storage. Many adenoviral vector vaccines, like AstraZeneca's, are relatively stable at standard refrigeration temperatures, 2 to 8 C, making their storage and global distribution simpler and less expensive than ultra-cold storage, -70°C, required for many mRNA vaccines. High production yield. Viruses like adenoviruses can be grown and purified in cell culture to achieve very high titers, quantities, facilitating large-scale, cost-effective manufacturing. Safety profile. The vectors are typically replication deficient, meaning they cannot multiply in the body or cause the disease they are based on. They also generally do not integrate their DNA into the host cell's genome, minimizing the risk of genetic alteration. Limitations and challenges. Challenge. Description. Pre-existing immunity to the vector. Many people have previously been infected with common viruses used as vectors, like human adenovirus 5, or Ad5. The body's pre-existing immunity can rapidly destroy the vector before it can deliver its payload, reducing vaccine efficacy. Requires rare or non-human vectors. To circumvent pre-existing immunity, researchers must use vectors to which humans have low exposure, example chimpanzee adenoviruses or CH-ADOX1, or switch vectors for booster shots, prime boost regimens. Vector-induced immune response. While the vector induces immunity to the antigen, it also generates immunity against the vector itself. This limits the effectiveness of subsequent doses or booster shots using the same vector type. Manufacturing complexity. Manufacturing involves growing the viral vectors in complex cell culture systems, sometimes adherent cells, and requires rigorous purification to meet high-quality standards, making the process more involved than chemical synthesis. Rare safety signals. Rare but serious side effects have been observed with some adenoviral vectors, such as thrombosis with thrombocytopenia syndrome, TTS, which requires specialized regulatory monitoring and clinical management. Limited genomic capacity. The vector has a size limit for the foreign genetic material it can carry. This constrains the complexity of the antigen or the number of different antigens that can be included in a single shot. Thanks for watching.